Okay, can you see the slides? Hello, can you see the slides?
Yes, sir.
Okay, so today let me start the presentation. Today I'm going to discuss on drug development, phases of clinical trials, and good clinical practice. You may be wondering why I should attend this class. Okay, so you may be wondering why I should attend this class. Because if you see the earlier question papers, in the last question paper, we asked that what is the importance of phase 3 clinical trials? What are the kind of tests we are going to do? So this question came into your question paper. Okay. And also in the last class, we discussed about sources of drugs. But we did not discuss about how to find new drugs. Okay, what is the need of the new drug? All these things. Okay, these things may also come in your question paper. And also there is something called as good clinical practice. So what is good clinical practice? This may be one of the question that may be asked in your theory examination, or most of these questions will definitely come in the practical examination. Okay, so in the practical examination we have spotters, that is related to drug development and phases of clinical trials. Various phases of clinical trials we will ask you in the spotters. Okay, there will be spotters related to phases of clinical trials and also drug development. Okay, in the drug development we'll ask about structure activity relationship. There are some spotters related to structures of penicillin, tetracyclines and adrenaline. So like this, some structures are kept in the spotters. So like this is a important this is a important topic, and I just told you about the importance of today's topic. Okay, so what are the single learning objectives which I am going to cover in this? Okay, I am going to discuss about the stages of new drug development, and also I'll explain you about the approaches to new drug development and how to invent new drugs. I'll talk about the preclinical studies, and also I'll talk about the phases of clinical trials. Okay, what are the different phases of clinical trials that are available, and also talk about principles of good clinical practice. Now what you are seeing here is the story of the Ramdesivir. So you may be wondering what is this Ramdesivir? Ramdesivir is a drug which was used for the purpose of treating COVID-19 disease. COVID-19 disease. You can see here there is something called as, it is a nucleoside analog. So you may be knowing that there is something called nucleosides in the DNA. Okay, DNA and RNA. For the preparation of DNA you require nucleosides. So if you fit wrong nucleosides and wrong nucleotides, then the DNA and RNA will be it's a fractured DNA or fractured RNA. It will be, means what I'm saying is it will not have a good standing of DNA or RNA, or its structure will get altered. So because of that reason, the proteins that are prepared inside that cell will be defective. Due to that reason, so the virus will not get replicated. So when Remdesivir tablet, this drug was invented, it was majorly invented for the purpose of treating Ebola virus disease. Okay, you can see that this drug was invented for the purpose of treating Ebola virus infection. But in the preclinical trials means before doing the testing in human beings. Okay, whenever we want to do development of a drug, at that time generally chemical testing will happen. After that animal testing will happen. So this animal testing phase is called as preclinical testing. Okay, in this animal testing, this Ramdesivir did not do good job. Okay, so it failed. Okay, so since it also did not do well in the randomized clinical trials, also in the human testing also they did, and at the time it did not do well in curing the Ebola virus. So it is a failure. Okay, Ramdesivir was a failure. So you may be wondering, then this failed drug, how did they use for treating what? Coronavirus or infection. Okay, but it was considered that it is a broad spectrum antiviral drug. Okay, it is a broad spectrum antiviral drug, and it was again demonstrated, it was demonstrating activity against ribonucleic acid viruses like RNA viruses, like various viruses were killed by this broad spectrum antiviral drug, that is Remdesivir, and it was affecting SARS-CoV COVID virus and MERS-CoV coronavirus, and then Nipah virus and Ebola virus also. It was inhibiting basically. So it was found that, okay, so empirically it was found that it is already evaluated and it is inhibiting some of these viruses. And as you know that coronavirus, that is the COVID-19, was a completely a new virus, and we were finding it difficult to find suitable treatment for it. So it was an emergency. At that time, they did few clinical trials, and as an exploratory trial, they continued to, like, as an emergency approval, the Remdesivir was allowed to get use in SARS-CoV, that's the COVID-19 virus, okay, for treating the COVID-19 disease. So like this, this Remdesivir came into picture. So you understood that what is the importance of a new drug. New drug, if you keep on developing at some time, like, if you have proper evidences, maybe an emergency like COVID-19, so at that time we were able to use many, it has been shown that it is being useful. It was useful, and it was found to be doing very good in the market. Okay, so you can see that within 6 months, from July to December 2020, almost almost 600 crores of money was made by this drug. But we don't always say that what is the success rate. It's not just by number of crores it made, it also how many people were saved. So basically at that time, since we were not having any other drug, so Remdesivir also, like, it was not very evident that Remdesivir was very effective in that, but it was found that empirically it was found that it is good, and it was inhibiting so many viruses, and they on the emergency basis they allowed it to be used in COVID-19 disease. By this time, now you know that it's very important for us to develop new drugs. Okay, so a lot of new drugs are required. So what are the conditions in which you require new drugs? Okay, so there can be limitations of some existing drugs. Okay, so at that time, like, so maybe some drugs are useful for kids, some drugs are useful for senior citizens, some drugs are used for only for adults between for 20 to 50 years, so like that. Or maybe like some drugs should not be used in pregnancy, or like some drugs should not be used in associate when the disease is in association with asthma, like, beta blocker should not be used in asthma, or some drugs should not be used in diabetes or in hypertension, or when two drug diseases are together. Like this, there will be some limitations when you are like developing some drugs. So if the drugs are already existing at that time, there will be some limitations. So to overcome these, you require new drugs. Okay, and also there can be chances that COVID-19 type new diseases will be invented. Okay, new diseases may come into picture, due to natural causes, or maybe due to human interventions, or maybe because of the environmental changes. So like this, new diseases keep on getting invented. So in that way, and also toxicity of the existing drugs. Okay, so many toxicities happens. Calcium channel blockers, they can increase the sodium and water retention, and also like beta blockers can aggravate the asthma. So like this, so some of the other toxicities can be there. Or so like chloramphenicol should not be used in children and also in pregnancy, so they can induce some teratogenic effects like that. Okay, so there can be issues like sedation. Okay, so when we use traditional antihistaminics, sedation is a major problem. Okay, so because of antihistaminics, and also anti-cancer drugs, they can produce lot of adverse drug reactions right from the top to bottom. Okay, hair fall to mouth ulcer to gastric ulcer to like organ failures. So like this, varieties of adverse drug reactions are produced by these anti-cancer drugs. So if you want to overcome this, then again you need to continue getting new drugs. Okay, so the existing drugs may have low response, okay, which is maybe negligible response, or if you keep on increasing the dose, that can get turned into toxicity also. Okay, so so whatever the amount of dose you are giving at that time, the drugs may not give the proper response, or like if you increase the dose, then there can be toxicity will be there. So in such conditions, again, you require new drugs which are more efficacious, better, safer drugs are required. At that time, again, you require new drugs. Then inconvenient route of administration. Okay, inconvenient route of administration. So what is happening here, like basically people are convenient taking oral drugs. Some drugs are not absorbed, like penicillin. Okay, penicillin, when it was introduced, it was not useful. Okay, it was not useful as a oral drug. Okay, so and again, insulin, even today we have the problem of route of administration, insulin. Many people don't want to take injections. Okay, so they want oral insulin. That is still not successful. People are doing evaluations, but still we are not able to get a suitable insulin to be that can be given orally. So little bit of success nowadays it is coming, still it is not successful. Okay, so oral GLP-1 analogs are available, but insulin, oral GLP analogs are for the treatment of diabetes. But there is some success is already been achieved there. So they are also proteins, but I think insulin, again, it's a protein, but we have not yet achieved 100% success. But hope that it will get realized. And also there are chances that when you give two drugs together, because two diseases are there, or disease is not getting controlled with one drug, so you have to add another drug. At that time, drug-drug interactions, some favorable drug interactions will be there, we don't have any problem. But sometimes you get unfavorable drug-drug interactions, then you will have the problem. So to overcome such drug-drug interactions, also we require new drugs. Again, patient complaints. Okay, patient complaints, patients are not accepting the drug, they're not taking regularly. Okay, because of some toxicity, because they are not comfortable, because the disease is not getting treated immediately, or there can be chances that it takes longer time to start the benefit. Okay, you keep on taking disease and also suffer from disease, and after you overcome, then you may get the response. Till then patient may not have some patience. Okay, so they may feel that okay, this drug is not effective, so I'll not take it. So like that, so if the patient complaints is not good, again you will not get good response. Then as I said, many drugs should not be given in pregnancy, pediatric age group, geriatric age group. Okay, so old age group, again early age group, so they are not taken in the, you can sometimes many drugs are not evaluated in pregnancy, pediatric and geriatric. So Generally, we don't know whether they are useful, they are good, or they are bad in these conditions. So again, you require clinical trials or drug development is required in these areas also, and also you require more safer drugs for these conditions. Okay, so you need to evaluate new drugs. And what happens over a period of time, you have old drugs, then there will be chances of getting the resistance. Okay, so especially antibiotics. Okay, so you have older penicillins, so they are not sensitive as of now. Okay, because what is happening, so organisms, microorganisms have developed some resistance, so they are not able to treat these diseases. Or cephalosporins, first generation, second generation, already organisms are not responding. Tetracyclines, again, they are not useful nowadays. Maybe because one is they are toxic, another is resistance is there. Again, it's like this. If you see, majority of the antibiotics, they are not treating these infections, because one, maybe because the people are not using properly, complete course is not completed, or there can be issues associated with that. So like this, and also environmental. Today is an environmental day. If you see that, I have attended one conference there. We learned that many poultry industry people, they are using antibiotics for better growth of the chicken or any other the animals, for the purpose of exporting meat. So like this, there are conditions. So for that, since they are using the drugs for that purpose, the drug organisms which are infecting animals, they will also infect us, or they may develop resistance there. Then they will transfer that gene to the other organisms. So all the organisms which are getting resistance there, they will get that gene will get transferred. That is a plasmid we call. So that plasmid will get transmitted to other microorganisms. Because of that reason, there will be drug resistance. So like this, lot of issues will be there. So for all these reasons, we require new drugs. Now, like, if in my viva I may ask you, why do we need to develop new drugs? Okay, so now you know that there are limitations to the existing drugs. New diseases are identified, toxicity to old drugs, low response to the old drugs, inconvenient route of administration of the existing drugs, drug-drug interactions, severe drug-drug interactions, or yeah, yeah. So then patient compliance, indications to, like additional indications like pregnancy, pediatric and geriatric indications. So and then drug resistance. So in all these conditions, we require new drugs. Okay, so hope all of you understood. Now you will write this answer in your answer paper. Now let me discuss about how to address these limitations. Okay, where do we find so many drugs? Like, so many problems are there. Like, with difficulty we find new drugs, and there are problems in these drugs, already existing drugs. Like, where do I find new drugs every day in, day out? So there will be sources. What are the sources of new drugs? We already discussed last time. So best way to find new drugs is you keep on researching, you keep on understanding the disease, you keep on understanding the molecular, the dynamics of that particular disease, physiology of the particular, person, particular organ, particular cell, and what are the molecular mechanisms involved in the either the derangement of the physiological system or development of a disease. What are the coexisting physiological changes that happens? If you understand all these, I think you will get some targets where you can treat that particular disease. Then how do we find new safer drugs? Like, so if you have some chemicals like insulin, it's a, like already existing in our body, so you study the structure of it and create a new analog of it. Okay, so insulin analogs, they are very useful. You can see that will be one of the main question you can get, or a short short note you may get in your main examination. Okay, insulin analogs. Okay, so the insulin is already there in our body. If you study the structure of it, you little bit modify here and there, so change some amino acids from here to there, then you will get new molecule. Okay, new molecule, and you are able to get some analogs. Some are getting faster absorbed, some are getting delayed excreted, and they can be used for longer time. So like this, you are able to get some new drugs. Okay, then derivatives of the existing drugs. Okay, existing drugs, you do keep on doing the permutation combinations of two drugs, and you may get one more new drug. Okay, and you exchange some groups. Okay, so at the suppose you have benzene ring at the position 1, 2, 3, 4, 5. So you keep on exchanging the methyl group, ethyl group, keep on adding some fat molecule, keep on changing the structure. In that way you can develop new drug. Okay, you can come up with the new structure, new, and you keep on evaluating. Then you do the some structure activity relationship analysis, you will be able to get a new drug. Then what you can do, if you want one drug is long acting, you can make it short acting, or you can like earlier drug was short acting, make it long acting. So in that way, by just adding or removing some some chemical groups or some chemical structure or structural alterations, you will be able to get that new drug. Okay, so this is what we call it as structure activity relationship. Okay, finding the structures and exchanging the groups within that molecule or adding new molecule, so you will be able to get new molecules. So this is how if you see adrenaline, noradrenaline, dopamine, they all look alike. Okay, only little bit of changes here and there. So in that way you are able to get all these active ingredients like dopamine, adrenaline, noradrenaline, all these like they are all derivative of the same molecule. Okay, so you can see that estrogen. Okay, so by like you can find some alternative to estrogen, that is raloxifene. Then morphine is naturally available. Okay, morphine is not naturally available. Okay, it's a product coming from opium, and it has a property of inducing constipation if it is used for, but it is having so many other problems. It can cause dependence and addiction, and like craving for the drug like that. So you can't use morphine for constipation. So if you use that, then there will be a lot of other issues, psychological issues and other issues will come in, and it will also reduce pain and addictions also. So like this, like a lot of other issues will come in. So you can use loperamide. It's a derivative of that morphine, and it can help in controlling the diarrhea. Okay, it's a synthetic product coming from that. So next thing is that what I'm going to talk is related to serendipitous development of new drug. You can see here in this picture, what you are seeing here is that, okay, so I think I explained in my last class related to identification of penicillin. So one day, as I said, in 1928, Fleming, he wanted to, he's a Scottish microbiologist and physicist. He wanted to go on vacation, and when he was leaving, he handed over some of the petri dishes to his friend. Okay, so he was growing some microorganisms in that, and he handed over, and he asked him to properly maintain and all, but his friend, he just left it as it is on the lab. And after 7 days or 15 days, when he came back, he saw that there is something called as this is the Penicillium notatum. This is the bacterial growth. Okay, so when this fungus is growing here, surrounding that there is no growth of the microorganisms. What you can see there is a gap here. Then he suddenly clicked into mind, why there is no growth here? Okay, why there is no growth here? And the growth of microorganisms, all these are microorganisms, the growth of microorganisms is inhibited by this. So there must be some chemical released by this Penicillium notatum. Okay, that's the fungus mold which was there. And then the famous discovery of penicillin happened just because. But actually, if you see, Fleming was doing research on something else. Okay, by accidentally he identified Penicillium notatum, and from there penicillin was extracted, and in that way serendipitously they developed penicillin. Okay, so you can see here, this is what has happened. Okay, so serendipitous drug discovery means they were researching for some other purpose, and they invented the particular medicine accidentally. Okay, that is what we call it as serendipitous drug discovery. If you see, most of the good things happen by accidentally. Okay, so always keep your eyes and ears open, and you will be able to find something interesting that is happening in your life just by accidentally. Okay, so that's what is happening here. Penicillin happened like that, and many drugs are happening like that only. Okay, so but they basically this is how. And you can see here, Paul Langerhans is a student, okay, medical student. He was interested in developing, he was doing research on pancreatic cells, and he identified there are two different types of functions, okay, that are there for this pancreatic cells. And in that way they identified alpha cells, beta cells. So beta cells were identified, and insulin was getting secreted by alpha cells. And Banting and Best, they did the work. Once it is known that insulin is available, and they know that that is treating the diabetes, and in that way they found that, okay, so insulin crystals were synthesized by Banting and Best. From there, like they used for treating diabetes. Okay, that's how the great invention of treatment for diabetes happened. Initially they used to do the crude extract of beef, pancreatic extracts and all, so they were giving those through injections, and then finally they were treating the diabetes. But later, like now we are doing getting the insulins through biotechnology. Okay, so in that way we are advancing our, like we have advanced ourselves and getting the purer form of the insulin. So now let me get into the next stage. What are the sources of drugs? You know that random screening, serendipity, then toxic plants. Okay, most of the drugs are toxins. Okay, so most of the drugs are toxins. In that way, like toxic plants or poisons, teratogens, like when so they can be very important sources of drugs. And metals and salts are the or the very important source, like iron, calcium, all these things are coming, sodium, potassium. So synthetic, so some of these synthetic sources are there. Recombinant DNA technology can be used for identifying the drugs. Modification of the structure of the existing drugs can be done for identifying the new drugs. Okay, so and also, like some of the drugs like Ramdesivir, which failed in the earlier research for something else like Ebola virus, but it was used for treating COVID-19 disease. Like this, like some drugs earlier, they failed for some reason, and you can try to work out the same drugs maybe for a better purpose, and you will find like this. Thalidomide, if you see, thalidomide was initially used for the purpose of treating vomiting during pregnancy, and that created the major thalidomide disaster. So because of that reason, what happened? This was banned. Basically, more than 20,000 babies they lost their, like when they are born, it is found that whoever the mother used the thalidomide. Okay, the mother if used thalidomide at the time, the babies were born without limbs, upper and lower limbs. So seal-like babies were found, and they were reported. Because of that reason, this major revolution happened in the field of pharmacovigilance. But later it was banned and it was kept in the shelf for longer time. But now recently, if you see, this thalidomide can be used in the purpose of treating lepra reactions in leprosy. Okay, leprosy patients, as a anti-cancer drug, in the treatment of anti-cancer drugs, there also as an anti-cancer it can be used for reducing the inflammation and many other issues, vomiting or adverse drug reactions caused by the cancer drugs. Okay, anti-cancer drug. Like like this, can you can go on. If you go back to the history, you will learn a lot that like old drugs are repurposed. Okay, you can see that in the COVID-19 we repurposed ivermectin for treating the, it's actually an antihelminthic, but it was used for treating the COVID-19. And chloroquine also, like in that way, if you see, chloroquine also it was introduced for some other purpose. Basically it is used for malaria, but now recently for preventing COVID-19 also it was used. So like this, like if you understand if you have the deeper understanding of the molecular mechanisms and also cellular mechanisms, so like this, if you understand, you will be able to find newer drugs. Okay, so you will be able to find newer drugs, okay, for treating various diseases. Okay, so I think I will continue in my so just give you examples. You can see chemical modification of penicillin led to amoxicillin and ampicillin, so which are given orally. Penicillin cannot be given orally. Amoxicillin, ampicillin can be given orally. Synthetic analogs and antagonists can be were developed for hormones, neurotransmitters, prostaglandins, and again, traditional medicines like physostigmine is a plant extract alkaloid, but neostigmine is a synthetic analog of physostigmine. Then fluoroquinolones are completely evaluated based on the structure activity relationships, and they were fluoroquinolones like ciprofloxacin and all, they are derived through the structure activity relationship, and antibiotics, as an antibiotic they are used. L-dopa is the another one. You can see that like L-dopa is a derivative of dopamine. It can be given orally. It's a pro-drug. Once it gets into the body, and once it enters into the central nervous system, it gets converted into dopamine and helps in the treatment of Parkinsonism. So recombinant technology, like newer technologies we are using for getting drugs like insulin, erythropoietin and all. Then single enantiomers, like every drug that is there as a chemical, they will have dextrorotatory form, liver rotatory form. So you can isolate one of this form, and you can see that the better potency of that particular drug, better availability of the drug, like esomeprazole. Omeprazole is there, it's a mixture. So if you find dextrorotatory form or liver rotatory form, so you will find like esomeprazole, it's a better availability, it is having better oral availability, and levofloxacin is having slower excretion, so longer activity is there. So like this, in the next class I will be talking about how to develop new drugs. Okay, so I will be talking about that. So today anyway, so we have spent sufficient time in understanding how to find the new drugs. Okay, so as I told you in your final examination, practical examination, in the Viva examination, you will be, asked about, so give some various examples how to develop new drugs, structure activity relationships. All these things may be asked, and also like your history of, penicillin may be asked. So if you learn about all these things and keep in your mind, so if you are asked about penicillin, you can write all these things what I just mentioned, how penicillin was found, so you can write about it. Then what are the, why do we need drugs, new drugs, how to develop them and all. So I think I just mentioned about all these things. Now I will end. Okay, now I will end. I will continue in my next class. It is related to how to develop new drugs, what are the phases of clinical trial. All these things I am going to discuss in the next class. Okay, so see you in the next class. Okay, is there any questions, any of you? 1 minute is left. Hello, Navya, who are all there online?
Yes, sir.
Any doubts you have? Did you
No,
understand?
sir.
Did you understand
Yes,
what I
sir.
was
Yes, sir.
speaking? Who was Akshay? Akshay, tell me,
Yes,
how
sir.
are
We
you?
understood, sir.
You understood?
Fine, sir.
Okay,
Yes, sir.
good. All of you are able to understand what I'm saying. Anu,
Yes, sir.
Lolita, Vinayak.
Yes, sir.
Okay, you are okay?
Yes, sir.
Okay, so let me end the class now. So we'll come back again. Okay.